And overall, it appears that taurine abundance associates with health status in humans, and low taurine abundance is associated with poor health in humans.
So, hello, and welcome to the Sheekey Science Show, where in this video I spoke with Vijay Yadav, Assistant Professor at the Department of Genetics and Development at Columbia University, researching functional connections between human metabolism and disorders. Earlier this year, his team published on the role of taurine and how supplementation in a mouse model improved many markers of health. Here we discuss Vijay's interest and research further. So, hi Vijay, thank you for joining me today and welcome to the Sheekey Science Show.
Thank you very much for the invitation. It is my pleasure to be on your show.
Thank you. So, earlier this year, you had a publication that caused a lot of interest for a lot of people on the internet about taurine and taurine metabolism and its influence on aging phenotypes. But before we get there, I'm actually even more interested in some of your earlier research that you did before getting to that stage, as I think it will also help us, I guess, understand why that focus and interest came to taurine. And so, firstly, I noticed that one of your earlier publications was about the corpus luteum and the regulation of apoptosis. And I was just wondering if maybe you could elucidate a bit on your early interest in science and the work that you were doing.
Sure. So my interest in doing research goes back to 19, maybe late 80s, when I used to go to my father's workplace. He was a finance person, but he worked in a science institute, which is a central drug research institute in Lucknow, in India. So whenever my mother thought that it was a vacation day and we had nothing to do and she wanted to get rid of us, she just put us on his vehicle and put us to his office so that she has some free time. Raising three kids is not a small thing. So so we just used to run around in his office area, not so much to do with the money, but because it was a science institute, we had exposure to going to laboratories. At that point the regulations were a bit less, so the kids used to think we are running around, the 9-year olds which are going around in the labs, you can imagine today happening. It is going to be a lot of regulations. So I remember one of my first interaction was that somebody was dissecting a cat. They were working on diabetes, and my question to the gentleman who was doing the dissection of a cat was, "What are you doing?" He said, "We are discovering medicines for diabetes." And that was my first question I could remember. Okay. Now, these initial episodes, I think, were very important because those were formative years, and that created an interest in me in research. And when I finished my master's in biochemistry, I did start my PhD in Indian Institute of Sciences in Bangalore, working on how female reproductive physiology is regulated by endocrine systems. And in particular, we were looking at the effect of ovarian physiology on the implantation process in the fetus. And when I started my PhD supervisor asked me, "What do you want to work on?" I said, "I would like to work on how menstrual cyclicity is regulated during fertile and non-fertile cycles in humans, and how can we help to understand this process better." He said that is a very poorly understood area. And I asked him this question way back in 1999, is that why it is a poorly understood area. He said that because if you want to really relate human physiology, you have to use monkeys as a model system. And well, I again asked him why. He said that because no other species on this planet undergoes menstruation like humans do, which we think are the highest evolved species on this planet, except humans and higher order primates, monkeys. I was not aware of these things because as a PhD student, you are starting, you are looking at physiology, but you are not aware of the intricacies of different processes. And he said that if you want to really work on this problem, you would have to use monkeys as a model system. And one thing I learned from him early on during my career was that if you want to address a biological question, you have to use the best model system available. And he told me, if you really want to do this, you have to really be prepared that your experiments are going to last 3 to 4 years, each experiment, and you have to overlap them so that you can finish your PhD in time. Well, for good, I took that challenge, and we started understanding how ovarian glands are formed and during the process of the menstrual cycle. And one of the transient endocrine gland in the body is corpus luteum, which actually is formed after ovulation happens, and it produces progesterone to help in the implantation process. It prepares the uterus for implantation to happen. And corpus luteum failure is one of the major problem for early pregnancy failures. And so during these course of the initial formative years, we use monkeys as models, and then we went back to cows as a model, bovid species, and rats as a model to try to see how evolution has shaped this process, and we evolved to a being which has this unique menstrual cyclicity, and that is changed during the fertile cycles. And during the course of these studies, I became interested in female diseases after menopause, because menopause is nothing but a failure of the ovarian system. Not a failure of ovarian system, I would call it. It is basically an exhaustion of the egg deposits in the ovaries, and therefore leading to menopause. And this happens around 45 to 50-year-old in humans. Now, these studies led me to understand female diseases after menopause, such as osteoporosis, diabetes, and obesity during my postdoc years. And I did not have enough training in mouse genetics at that point to be able to mutate genes, to be able to do conditional mutagenesis, to address physiological questions. And during my postdoc, I worked on osteoporosis, obesity, and how appetite is regulated in all these energy demanding processes. And after I finished my postdoc, I started looking at how different nutrients and these processes are affecting the processes of aging, because all these processes get clustered and they get together at the end of our lives, and we start experiencing this morbid period, which is pretty long for a human being. And in order for us to compress this morbid period at the end of our lives, we need to understand this aging process better. And that is where I started my independent career, looking at different nutrient molecules and different genetic factors which predispose us to these aging diseases. And that is what led me to, in the initial years, led us to discover the importance of vitamin B12, an essential nutrient coming from animal based diet only, in regulating human physiology. And that led us further to a role of taurine in regulating various physiological problems or various physiological phenotypes, such as growth and bone density in early part of our lives. And that is where we started to address this question of the wider role of taurine across organ systems, and we stumbled upon the effect of taurine during the aging process.
Wow, that's a lot of experience, and awesome how they all sort of followed on from each other. Because you mentioned the relationship about vitamin B12 and with osteoporosis, but generally, what is known about vitamin B12 and how is it used within cells?
So vitamin B12 is a very interesting molecule. It is one of the most complex vitamin of all. In fact, it has a very interesting story. Till about 1910, there was a disease known as pernicious anemia, and there was no cure for it. And then a scientist created this animal model whereby he was bleeding dogs and feeding them different diets, and trying to identify, is there a dietary component to pernicious anemia. And when he bled these dogs and he fed them animal based products like liver, raw liver, and muscle, their anemia was reversed.
Hmm.
So he noted that perhaps there is something coming from the liver that is a very important molecule or a factor that is regulating the anemia process. Based on these initial findings, without knowing what is present in liver that is curing anemia in dogs now, two clinicians decided to feed humans with raw liver. So, something like 500 grams of raw liver served to you on a plate in the morning. You can imagine what your response is going to be. But people were dying from this disease. That is
Hmm.
why it is known as pernicious. So, they had no option. Very surprising thing that these clinicians noted, one in Canada and one in the US, that when they fed humans with raw liver, their anemia reversed. And within 4 to 6 months, they started walking and running around.
Wow.
From the phase of immobility, they were able to now walk and run around. I had this wonderful video which was published in Blood Journal, and I was trying to look for it for some time, but I have not been able to find. But that shows how a bedridden person with pernicious anemia started eating raw liver, and then he starts to walk, then he starts to run. So
Wow.
this, till till that point, till about 1930s, we had no idea what was in the liver that was curing pernicious anemia. Well, three people got Nobel Prize for it, for feeding humans raw liver and curing pernicious anemia: Whipple, Minot, and Murphy in 1934. And that is the greatest example of how important it is to really identify cures of a disease without knowing, in fact, sometimes what it is that you are doing that is curing the disease. Well, 20 years later, then Dorothy Hodgkins in the United Kingdom, then she actually identified the structure of vitamin B12, got a Nobel Prize for it again. Um, and it also elucidates that, okay, we, from the point of a basic scientist, clinicians, and then a structural biologist, how we were able to identify a molecular origin of a disease. And till that point, there was only way we could synthesize. There was no way to synthesize B12 till 1950s. Then these labs in one in Harvard, I'm skipping the name, Arzenmauer in Switzerland, and Woodward, if you can correct me if I may be wrong there. Woodward in Harvard, they synthesized B12 chemically, and it took them around 15 years to do so. In fact, Woodward's lab was burned down completely during the process once,
Wow.
because it was so complex a molecule. They had to collaborate with the Swiss labs to really see the precursors, and then going back and forth to be able to do so. At the end, they were able, I think, in sometimes in late 1960s, they were able to synthesize completely the B12, and after that, we have the synthetic B12 available. Anyway, I think it was a bit of a long route. You were not probably expecting me to narrate, but this is how the B12 came in picture, where you have this disease, molecule gets identified, and then you have you have it available to people to cure pernicious meaning anemia. And later part of our lives, we do have this vitamin B12 deficiency. A major chunk of us go through this B12 deficiency phase when we are 60-year-olds. And this episode also illustrates the importance of nutrients coming from specific dietary origins, which are important for our health. And perhaps it is a time to better understand what nutrient molecules are actually required for our health. We have barely any idea of what are the nutrients which are essential or semi essential for us for a better health.
Yeah. That was firstly a super interesting story, but also I think you're completely right. I mean, there still is, even today, when people talk about health, no one, people tell you take too much vitamins or not enough one, and it's not just vitamins in general; it's different types of vitamins, as you just mentioned. And so, with vitamin B12, is it mainly through diet that we get it, or to what extent is our bodies capable of naturally producing it?
B12 cannot be produced by human body. It is only, well, the reason for that is that only some bacteria in animal kingdom can synthesize B12. And these bacteria need to be present in earlier part of the GI tract, such as in ruminants. There is a pouch before the stomach, which can house the bacteria, and bacteria can synthesize it. And then when it passes through the stomach and the intestine, there is a protein produced only by the stomach cells, known as a gastrointrinsic factor, that binds to B12 and that helps in the absorption. So if B12 is synthesized in a later part of the GI tract, it cannot be absorbed. It can
I
only
see.
be absorbed if it is present before the stomach, and that ruminants can do it. And when we eat these muscle products or liver or animal based diet, we are able to obtain B12 from these diets. And today we can do it synthetically as well.
I see. And so is it still unclear as to once you've got vitamin B12 in the body, what it's interacting with, or how is it actually functioning in the cell?
And it has been a while since B12 was discovered, and people have worked on a variety of different molecular processes within the cells. Only two known enzymes are requiring B12 in our cells. That one is methionine synthase, and other one is MUT, which is present in the Krebs cycle. And these two enzymes are requiring this B12 as a cofactor, modifications of B12 as a cofactor. And then downstream of these processes, you have a variety of changes happening in the cells, which is still at a cellular level, at a metabolic level, we do not understand precisely. And that is where we stumbled upon a role of taurine, because we were trying to figure out how B12 is affecting the growth and bone metabolism in animals. And when we so we were noting there was a decreased osteoblast function in these vitamin B12 deficient animals. But when we put B12 on top of these bone cells, nothing happened to them.
Right.
So we thought, okay, there must be some endocrine origin of this B12 action in these animals. And B12 is, as the pernicious anemia episode tells you, it is predominantly stored in liver and to an extent in muscle and recycled in the body. And so we did a metabolomic analysis in B12 deficient livers. Way back, I was in Cambridge that time, and we identified that through metabolomic analysis, that B12 deficient livers have a variety of downstream processes that are perturbed. And through machine learning and other methods, we could then narrow it down to processes which are critical for B12 deficiency to precipitate its effect on body systems. And we identified that in B12 deficient livers, there was a dramatic reduction, around 95 to 98% reduction in taurine levels in these livers.
Wow.
And if you supplement B12 deficient livers with taurine, you are able to help their organ functions. And that led to the identification that B12 regulates taurine biosynthesis, and through that means it is able to affect in an endocrine manner a bone deposition and growth phenotypes in different cell types.
Interesting, because so with taurine, I know that we can get it from diet, and I think we'll come on to that shortly. But in taurine's case, it can be synthesized in the liver cells, if I'm correct. And so what you're saying is that there seems to be maybe upstream a link between vitamin B12 and regulating this generation of taurine within liver cells. But you mentioned about the Krebs cycle, and obviously, I know that taurine is can be synthesized from cysteine, which is an amino acid, unlike taurine, which is a free amino acid. But then amino acids are sort of can be generated off from the Krebs cycle. I mean, maybe not cysteine. My biochemistry knowledge I should be better than this, but yeah, would you be able to elaborate more on or speculate on maybe how you think these two are related?
Yes, so the amino acid metabolism or Krebs cycle, as you mentioned, have an intricate relationship because many of the amino acids are metabolized through Krebs cycle,
Hmm.
through variety of intermediates. And although Krebs cycle is classically known to be associated with the production of energy by producing these NADH molecules in mitochondria, it also has a very important role to play in anabolic reactions. So when it comes to energy production, is a catabolic reaction. When it comes to different intermediates being produced by the Krebs cycle, there are anabolic reactions as well. And cysteine is one of the known precursor of taurine in the liver cells. Um, for taurine to be biosynthesized in the cells, we do have a limited capacity to synthesize taurine. And it is a free amino acid, as you mentioned, because it has a sulfonyl group in place of a carboxyl group, which is required to make peptide bonds in proteins.
Hmm.
So it remains
Yeah,
to the most part as a free amino acid. And taurine is interesting in many regards because its known functions are related to senescence, related to cell proliferation, and so on. But having said that, cysteine is the precursor of taurine, there are other means to synthesize taurine as well, and that we are stumbling upon now. And what are the critical requirements for different molecules? Like you mentioned, B12 is an upstream regulator of taurine biosynthesis in liver cells. And very interesting, B12 is stored in the liver, taurine is produced in a major way in the liver. However, during the course of our lifespan, there are phases in which we cannot produce taurine, such
okay,
as in during the perinatal period, babies cannot synthesize taurine, and they're dependent from mother's milk to obtain that taurine. And that is why many of the formulas that you see in the market today are supplemented with taurine
right.
without knowing what it is doing. Only thing people have predicted that it may have some role to play in the growth of uh, babies. And during the later part of our life, we start developing some ability to a limited extent to synthesize taurine in tissues like liver, in pancreas to an extent, in brain to an extent. But many tissues are deficient in taurine production, and they're dependent on the dietary intake. In humans, I'm talking about dietary intake as well as limited amount of taurine being synthesized from these different organs, such as, for example, muscle cannot synthesize taurine at all. And therefore, the first symptom that one sees in taurine deficiency, it is actually muscle wasting and neurological symptoms.
I see. And so, in terms of how we can get taurine then from a dietary, we need this a transporter present as well, right, to be able to take that into the cells. As I believe that there's a mouse model that's been generated to sort of mimic the absence of the dietary intake of taurine, and that was by inhibiting the transporter. But so that's the only transporter to get taurine into cells.
It is not. but that is the
Okay.
predominant transporter. So SLC6A6 is the predominant transporter which is required on the cells for taurine uptake. Because when you knock out this transporter, um, majority of the tissues have this taurine deficiency. However, this is not the only transporter. Taurine can also be exported from the cell by voltage regulated and ion channel. It has many other transporters which are present on neuronal cells, for example, on intestinal epithelium, for example, for dietary absorption. And therefore, that is not the only transporter, but that is one of the dominant transporter required for taurine to be taken into the cells. And that
I
is
see.
SLC6A6.
I see. And so when I read your paper earlier this year, I was trying to find out more about taurine, not just how it's metabolized, but also like what it's actually doing within a cell. And it seems to be doing many different things within a cell, as you mentioned a few already. But from that to like mitochondrial translation, all the way to like bile acids and fat emulsification. I was wondering, yeah, how much is really known about what taurine is actually doing within a cell?
So there are many processes which have been associated with taurine, such as regulation of osmosis. Because it's a free amino acid, it has a charged amino group as well as a sulfonyl group. So it is functions as a deuterion in the cell, regulating osmotic pressure in the cells. But its concentration ranges from somewhere in micromolar range to millimolar range.
Wow.
So, two tissues like muscle, for example, and retina, for example, are very high in taurine. And it is required for their functioning because in taurine deficiency, you have muscle wasting and retinal degeneration in humans. So, humans with the mutation of this transporter SLC6A6, for example, have retinal degeneration and have a poor eyesight uh, in humans. But there are also animal models, such as cats, for example. Cats cannot produce taurine at all during their life cycle. And because they cannot produce their taurine, if their diet is deficient in taurine, you have this precipitation of growth defect and blindness in cats. In fact, that is how taurine came into limelight.
Oh, interesting.
So, almost taurine was discovered in 1827 by two scientists, Tiedemann and Gmelin, but it remained like a ambiguous molecule for a very long time, almost 140 years. Nobody cared for this molecule because biochemistry and molecular biology was just evolving. And in 1950s, there was this increased demand from society to provide pet foods. And when these pet foods were provided in form of pellets to the pets, there were many diseases that precipitated, like growth retardation, diabetes, obesity, and so on. The most striking aspect of this episode was actually development of blindness in the cats. So you can imagine these rich households having these cats which are going blind, hitting walls and dropping vase
Hmm.
and whatnot. And that was initial impetus to really identify what is in the diet that is actually absent, that is causing the blindness in the cats. And from 1950s to almost for 20 years, scientists trying to figure out what is missing in the diet. And they identified in a seminal paper published in Science in 70s that retinal degeneration or blindness in the cat is caused by a single molecule deficiency in their diet, and that molecule is taurine. And since then, taurine has, meaning cat diets have been supplemented with taurine, including dogs as well now.
I see. Wow. Yeah, that's super interesting. And so, as well, with your most recent study, you noticed that when we age, there seems to be a decline in taurine levels, and obviously there are going to be some tissues, therefore, that might be more susceptible, as you've already mentioned, maybe the eyes and the retinal tissue and muscle tissue. And so, does that then suggest that the decline is more likely due to dietary issues, or do you think it's due to the fact that the liver can't generate as much as we age?
Yeah, so I would classify that as taurine function during aging. And taurine function decline during aging, we have noted that this is due to three different events. One is the taurine uptake being compromised from the intestine. Second is taurine not able to act within the cells, and there is a kind of taurine resistance in some tissues. And third is that inadequate taurine being produced by the tissues that actually are having a limited capacity to synthesize taurine. So all these three events lead to functional taurine deficiency in animal species, and I would suspect that is going to be the case in humans as well. However, I should point out that we do not have a good understanding of taurine metabolism in humans as of now, especially during the aging process.
Hmm. And so, in your paper, once you observed this, so was that the initial observation, the decline of age, that suggested that maybe we should try supplementing it in the mouse models that you used?
Uh, well, this was the initial impetus. In fact, initial impetus was when we noted the B12 deficient animals having less taurine, having diseases of aging such as osteoporosis, anemia, and cellular functions which are known to be affected during the aging process. But then we started looking at, okay, does taurine affect the aging process? Because that was not known when we were starting way back in 2012. And we measured taurine levels in different species, and we noted that the taurine levels also tend to decline during later part of our lives, especially when you compare with a young age. In fact, if you compare with fetal tissues, there is more dramatic decline in age tissues. So fetal tissues compared to adult tissues have something like 5 to 10 fold higher taurine levels
Oh wow.
without
Okay.
us knowing what it is doing in fetal tissues. So, and this decline in taurine levels led us to then investigate in mouse. We started and to do the intervention study where we did the middle-aged animals which have low taurine levels. We started feeding them taurine once daily after optimizing the dose and frequency of this intervention. And that is what we noted, is that taurine intervention led to increase in the lifespan of these animals, and not only lifespan, it also increased variety of health parameters in the animals. In other words, health span also was increased, and the morbid period was suppressed at the end of our lives in animals. And that was an initial study that led us to then investigate across evolutionary spectrum, including in worms, in the yeast, in monkeys, and some experiments in zebrafish, and finally in humans to see what taurine is doing during the aging process as far as the health is concerned.
Exactly. And so in the paper, I believe that it wasn't sex specific, it was beneficial for both the male and female mice. But was there a difference in terms of how effective it was that you noticed?
I think the in females responded a bit better, and there are these sex differences that are also noted in other interventions. Sometimes females respond better. Often, actually, females respond better, and males to a less extent. And that has something to do with, in my view, uh, the reproductive cost of the aging, um, because reproduction and aging are tightly linked, and species that invest too much energy on the reproduction have perturbations in the aging phenotypes. And so there are sex differences in aging interventions that also depend on what are the molecules being produced in females versus males, and something to do with these ovarian cyclicity that gets compromised at later part of our lives, and in most species as well.
I see. And so, when you looked at the different benefits to their health in terms of things like bone density on the taurine supplementation, were there so what were like the most shocking or most interesting findings that you found?
Um, to me, the most interesting aspect of these entire thing was that these animals were less obese. Although their body weight was not dramatically reduced in taurine supplemented animals, their fat deposition in abdomen and other tissues was dramatically reduced. And these animals had taurine supplemented animals had a dramatic increase in energy expenditure. So you can imagine that now these animals are the same except they are having taurine supplementation, and they have this enormous burst of energy expenditure for which I don't know what is the reason behind it. I would like to know one day, hopefully. Um, because their diet, the food intake was not dramatically perturbed, and they were depositing less fat because they were burning more fat.
Hmm. That's
And that, I think,
interesting.
is very interesting phenotype I would really want to know, because if we can figure it out, we have a means to, for a major human population which doesn't want to exercise, to reduce obesity.
Yeah, no, that's super interesting. Because I mean, when you initially said it, it then made me think that maybe taurine then, when you increase the supplement, the amount being supplemented, is something to do with the bile and the emulsification of fat. But then in many ways that would in theory increase fat absorption. I mean, I'm not entirely sure on how it all sort of works, but instead you're seeing increased energy expenditure, which then posits to maybe better mitochondrial functioning or something linking with the translation. I'm not sure, speculating here. No, that's definitely interesting. But as well, maybe things like the uncoupling proteins, because I know that like with the mitochondria and the electron transport chain, if you have like the heat production, so the uncoupling protein, so brown fat is what I'm trying to get to. Maybe that might be a link. I don't know.
Yeah.
I guess
There
yeah.
is an increase in the UCP1 expression in taurine supplemented animals. And UCP1, you pointed out clearly, that has an important role to play in mitochondrial respiration. And there is a increased abundance of UCP1 in taurine supplemented animals, especially in the brown fat that we noted. And that could be one way it is actually involved in heat production in the body. And perhaps one way these animals have more energy expenditure.
Ah, interesting. Okay. I feel
You are on the point.
happy. I think that was kind of cool. So as well in the study, you also so obviously you're supplementing the mice, and I don't know how necessarily that would translate to body like per kilogram to humans, because obviously humans we can get it from things like energy drinks and obviously meat products. But what sort of levels are people normally getting from diets in terms of taurine on a day-to-day basis, and how would that compare to what you gave the mice in the study?
So the dietary intake is less, because the highest amount of taurine from the diet, it is present in shellfishes, which is approximately 1 milligram per gram of shellfish. So even if we eat, let us say, 500 grams of shellfish, we get 500 milligrams of taurine, and that is not the case.
Okay.
Um, Now, coming back to what is the human equivalent doses that we used in different species. So, different species, for example, in mice, we use 500 milligram per kg body weight or 1000 milligram per kg body weight. The monkeys, the dose translated to approximately 250 milligram per kg body weight. And if we now look at the human equivalent doses, there's a formula that converts mouse doses to monkey doses to human doses, which takes into account surface area, km values, and etc. Human doses would be around 3 or 6 gram per day of taurine for a 80 kg human being.
Hmm. Okay, I see. And so, the paper that you showed, the research that you showed in the publication was that based on like dietary intake, I think there was like an association with the amount of taurine present. Actually, I can't quite remember what was in your paper. That's quite bad. At the very end of your paper, you showed some human data. What was the finding from that part?
So all these species in investigations from worms, mice, zebrafish, monkeys led us to think um, that taurine deficiency is a driver of aging in these species. And the next question we addressed actually was what happens in humans.
Hmm.
And so we had access to these 12,000 subjects. longitudinal health data, which was available from Epic Norfolk study. And we then performed a clinical correlation or clinical factor correlation with taurine and its metabolite levels in their blood. And what we identified is that if these aged humans, which are like 60-year-old humans, they had a higher taurine levels, they had a lower obesity, lower BMI, lower waist to hip ratio, less prevalence of type 2 diabetes, less hypertension, better glycemic index, less liver damage, less inflammation markers in the bloodstream. And overall, it appears that taurine abundance associates with health status in humans, and low taurine abundance is associated with poor health in humans. Conversely, in humans, we also investigated what will happen to taurine metabolism in a healthy state and what could be a healthy state of a human being than exercise. So we put these sedentary individuals, marathon runners, bodybuilders, and sprinters through a bout of exercise. And this bout of exercise in these individuals led to increased abundance of taurine and its metabolites, irrespective of the kind of individual we put them through exercise regimen. And that also tells you that, okay, low taurine abundance in aged humans is associated with poor health, and now a healthy state of a human being is associated with a better or more taurine abundance in their bloodstream. And perhaps some of the health benefits of exercise are mediated by taurine abundance in the bloodstream.
And so, given these findings, am I right in thinking that you're now hoping to set up human clinical trials to test the efficacy of supplementing with taurine?
Uh, you're absolutely right. We are putting together a multinational trial where we are trying to figure out. No, let me go back. So we still do not know how taurine metabolism is going to change from one ethnicity and one population to the other.
Hmm.
We only have tested in one population the changes in taurine and clinical variables, right? And one could imagine taurine being coming from a diet based or coming from a diet will have a very strong changes when you look at a population that remained omnivore or that remained that became due to development of agriculture more grain based diets. So this evolution of dietary genetic interaction is still need to be investigated, especially when
Yeah.
it comes to taurine. Um, because and if you look at the human history, the modern human history is 200,000 years, right? And you can see that in the most of it, this 200,000 years, we have been hunter gatherers in the wild. Only in the last, let us say, last well, I would say 1200 years or so, we have now become increasingly domesticated, or we can call it we have imposed self domestication on ourselves. Not only we impose self domestication on ourselves, we also domesticated species.
Yeah.
And if we have to learn from this entire episode, one thing we can learn is that when we introduce these diets, specific diets to these pets, they developed different diseases, cats and dogs. And therefore, we need to first understand what is the requirement depending on the genetic makeup of individual which have migrated out of Africa a long time ago towards northern hemisphere or southern hemisphere and developed different dietary patterns. Perhaps genetics and nutrition has co-evolved together and shaped this evolution process. And we are still trying to understand that what is the different or what is the reference range of taurine in different population, and also to do in humans a intervention study which we hope to start hopefully by mid to late next year, uh, where we are going to investigate the whether taurine intervention delays the pace of aging in humans, across different populations.
Nice. As and so, what are sort of the markers that you're hoping to look at? Because obviously, a lot of markers are doing things like DNA methylation, but obviously, in this case, there's a lot of the looking at the bones density. There's a lot of other things that you might want to consider in this case, as I feel like things like sarcopenia or blindness as well are like very interesting things that seem to be uniquely tied to taurine supplementation as well.
So we want to look at the health span of the human being, 6 months to 1 year after taurine supplementation, as well as we want to look at different hallmarks of aging in their bloodstream, such as, for example, immunosenescence, looking at methylation clocks, looking at how their organs are aging in different through different analysis of different markers in the bloodstream, and try to then put together an analysis that can tell us whether taurine supplementation delays the pace of biological aging. And taurine is not the only molecule that is going into taurine to intervention trials now. Meaning, there are increased interest in rapamycin, metformin, and these are lead runners molecules which are going to go to intervention trial hopefully soon. And we will have probably in next 3 to 4 years this anti-aging basket full of these molecules, which probably will guide precision medicine in next 5 to 10 years, I would suspect.
Yeah, no, it's super exciting times at the moment. And so with taurine, one other thing that obviously happens when we age is we have declines in our immune system. Is much known about the link between taurine and immune function.
Well, a lot was known before we started even. In fact, taurine has been shown to be important for quenching these oxidative species that are generated during this oxidative burst required for immune cell activation. And if you look at PubMed, you will find molecules like N-chlorotaurine or taurochloramine that actually are formed in immune cells by combining hypochlorite, a reactive oxygen species generated in immune cells, which has the potential to damage the immune cell themselves. But then taurine comes in picture, it binds to hypochlorite and makes taurochloramine. So taurine is very important for immune cell function. And it has been classically in the 90s, there were a lot of studies which looked at taurochloramine as a molecule, as a potent antioxidant in the antioxidant process, especially when it comes to the oxidative burst of immune cells.
Interesting. And so, is most of your lab at the moment focused on taurine, or are there any other interests that you're investigating at the moment?
So we are looking at different nutrient molecules, how dietary composition, how these nutrients shape up the genetic makeup of individuals, and how genetics informs the nutrient intake at the same time. To really identify, as I said in the beginning itself, the domestication of a species, including humans, were beneficial for many things. For example, we were able to cure many diseases which were like infectious diseases and so on. But then it also precipitated many other diseases which were caused by this nutrient genetic imbalance. So if we can identify these nutrient factors or the molecules which are required for our in our diets which can affect the health, perhaps we have one way or simpler ways to really benefit health without going to pharmacological drugs even. And that is one major interest that I have. And we continue to explore other molecules which can participate in regulating cellular health.
Nice. So what's been really nice about talking with you today is I love the way that everything you've done throughout your career so far has kind of like been a step onwards from what you've done before. Like there's been a logical connection. So as a student myself, and I know some students also watch, I just curious to know if you have any sort of advice for being a scientist or any sort of lessons that you've learned throughout your career so far.
Well, I think one of the most important lesson I have learned is that you need to follow your passion. There are going to be a lot of ups and downs in your career trajectory. Well, mostly downs, but one has to keep your enthusiasm alive. do bit by bit, slowly continue to build your knowledge, expertise, and address important question. And as long as you're passionate about addressing a question, you will find an answer. May not be the perfect answer, because biology is a fluid process which keeps on evolving, and our knowledge keeps on changing as we keep accumulating new information through new technologies. But at the end, it is just follow your passion and you will find your way.
Exactly. And also was curious about, you have an extensive knowledge about the history of the research that you're doing as well, in terms of like the earlier papers that were published. And I was curious to know, I mean, how do you go about like keeping up with literature or reading older literature? Is it mainly based on papers, or do you read much books, or do you have any book recommendations for my viewers?
I actually was a bookworm back in my PhD. So I used to read
Nice.
a lot of these old journals. I think that is no longer a uh, exciting thing for current generation, including myself now at the moment. Um, because we tend to believe in a lot of knowledge that we gain by just Googling information. And Google can be a very informative venue to look information, but at the same time you need to be knowledgeable enough to fish out right information from the wrong information, because
Yeah,
everybody seems
that's
to
true.
have an opinion today for anything that comes in any field. For example, people can have opinions, and science does not run by opinions. It runs by experimental data and facts generated by hard work, which people put through. You can imagine people working day in and day out trying to figure out some problems. But having an opinion is easy to have, but at the same time we need to be considerate of the literature that is present, go back to the original studies that actually established those founding principles, and that also elucidates how these technologies have evolved and what can we do better going forward as far as technologies are concerned.
No, I like that. I think we share the same mindset. So obviously, I'm blessed somewhat living in Cambridge, UK at the moment, which has like so many nice old bookstores. So I've accumulated quite a few old books myself, and I think the other reason why I say it's a wise idea is that, as you mentioned, we Google things nowadays, but obviously sometimes a lot of old books there's not PDF versions, or there's not an easy way of that information being put online. So you can't find it unless you actually go to the source and actually read it. And yeah, with current experimental improvements, maybe there are things that were suggested that we can now actually test and do more rigorously. So I'm going to agree with that mindset.
Well, thank you. I think I think we have the same point.
Excellent. So, no, it's been great speaking with you today, and I'm really looking forward to hearing more about the trial with taurine, as everything sort of happens, hopefully next year. And it's been fascinating to hear about your career trajectory, and yeah, I wish you just the best of success for everything going forward.
Thank you very much, Eleanor. It was a pleasure being on your show, and well, it is always a learning process, and I am delighted. Hopefully, people will see this experience and take it forward. Science.
Thank you.